this video I'll be solving paper 2 variant 1 May-June 2023 AS level biology. Starting off with question number 1, where figure 1.1 is a transmission electron micrograph of a cell from the stem of a sago pondweed, and they've given you the name here. So let's start with transmission electron micrograph. This is an image that's produced from a transmission electron microscope, which allows you to see inside cells, as opposed to a scanning electron microscope that only scans the surface of the structure. Okay, let's see what we have to do here. A part 1 asks you to state the evidence from figure 1.1 that shows that the cell is from the stem of this plant and not from the mesophyll of a leaf. So the difference between cells from any other part of the plant and mesophyll cells is that mesophyll cells will always have chloroplast. So inside of a plant cells, the only two types of cells that do have chloroplast are the mesophyll cells as well as guard cells that you find around stomata. For now, that's what we are going to use to distinguish. So here you don't see any chloroplast in the electron micrograph, which is why we know that this is not a mesophyll cell. Part 2, complete each row in table 1.1 to identify a cell structure shown in figure 1.1 that carries out the function listed. So gas exchange occurs through the stomata in a cell surface membrane. Now here you can identify the cell surface membrane for that. After that, you have the production of subunits in ribosomes. Those are produced by the nucleolus, which is this over here. So you see this boundary this is the nuclear membrane, inside of which you have the nucleolus, and then we have the function of active transport of ions. This also occurs across the cell surface membrane over here using channel proteins that are found inside the cell surface membrane, followed by aerobic respiration that occurs inside mitochondria, and that's the structure over here. So that will be E. B, plant vacuoles develop when vesicles fuse together. So vacuole is the one large body formed from various vesicles. The vacuoles increase in size as more vesicles fuse. Figure 1.2 shows the movement of vesicles within a plant cell during the development of a vacuole. So here you can see different processes. At X, you have movement of substance into the cell, and then you see this vesicle fusing with a larger body. You also see a vesicle budding off of the rough endoplasmic reticulum, moving towards the Golgi body, and then vesicles moving from the Golgi body towards the plant cell vacuole. Here you can see that you have keys for protein folded correctly and not folded correctly, represented by a triangle and a square respectively. Part 1 asks you to name the process that's occurring at X. So this is movement of substance into a cell through vesicles, and so this will be called endocytosis. Part 2, some of the vesicles formed by the Golgi body pass to the vacuole. These vesicles contain proteins that have been folded correctly and some that have not been folded into their correct shapes. The proteins that have not folded correctly pass to the vacuole where they are broken down. Okay, so they are not folded correctly, which probably means that they don't have the right interactions that a protein 3D structure has, or a tertiary protein structure has, and this will be corrected by processing in the vacuole. Explain how proteins that have not folded correctly are broken down in the vacuole. To break down a protein, you would have to break down the most fundamental part of the protein, So as you know, protein is a polymer that's made up of amino acid monomers, and these amino acid monomers are connected to each other by peptide bonds. And if you want to break down a protein, you would have to break down these bonds by hydrolysis using a protease enzyme, as you're breaking down a protein, and the respective enzymes for hydrolysis of proteins will be called protease enzymes. S. pectinata may have roles similar to lysosomes in animal cells. So lysosomes in animal cells are used for cell autophagy. What this means is these vesicles will engulf particles that are foreign or pathogenic, and they will digest them into non-harmful products so as not to damage the cell. And here apparently these small vacuoles play the same role. Describe the role of lysosomes in animal cells in defense against pathogens. So these pathogens in animal cells are engulfed by phagocytes through phagocytic vesicles. Phagocytes are one of the two major types of cells in the immune system that fight against foreign particles or pathogens. Once phagocytes have engulfed these pathogens through phagocytic vesicles, lysosomes will fuse with the phagocytic vesicle. So let me just try to draw that over here. Let's say you have this phagocyte, and budding in from the cell surface membrane is this vesicle which is starting to form. This is the phagocytic vesicle, and once this has entered the cell, it will then fuse with lysosome. Because this vesicle on its own can't do much, it will utilize the help of lysosome, which contains lysozyme enzymes. These enzymes will then digest any pathogens that were engulfed by the cell into harmless products. Question 2. Glycogen and cellulose are polymers. So glycogen is the storage polysaccharide found in animal cells, mostly in liver and muscles, whereas cellulose is the structural polysaccharide found in cell walls of plant cells. Figure 2.1 shows small representative regions of a glycogen molecule and a cellulose molecule. Okay, so here you have a glycogen molecule where you can see 1,4 glycosidic bonds as well as 1,6 glycosidic bonds. What this means is you have a glycosidic bond between the first and fourth carbon atom, as well as glycosidic bond between the first and sixth carbon atom between two glucose molecules. So here's how you number them. You have 6 carbon atoms in any glucose molecule. The 1,6 glycosidic bond forms the link or the branch, whereas 1,4 glycosidic bond results in the straight molecule. In cellulose, on the other hand, you can see only 1,4 glycosidic bonds. So there is no branch in the cellulose molecule. A. Describe 3 ways visible in figure 2.1 in which the molecule of glycogen differs from the molecule of cellulose. Okay, one other major difference between glycogen and cellulose is that cellulose molecule have a different building block. They are made up of beta glucose molecules, whereas glycogen is made of alpha glucose molecules. So you'll see that the hydroxyl group in alpha glucose molecule on the first carbon atom is projected downwards or away from the oxygen atom that you see in the ring. However, in a beta glucose molecule, the hydroxyl group on the first carbon atom would have been found pointed towards the oxygen atom in the ring, which is used to form this glycosidic bond. Okay, so the three differences can include the fact that cellulose is made up of beta glucose molecules, whereas glycogen is made of alpha glucose molecules. Beta glucose has unbranched structure, whereas glycogen has a branched structure due to 1,6 glycosidic bonds that connect the glucose molecule together. On the other hand, cellulose molecules, in the cellulose molecule, the adjacent glucose molecules are joined together by 1,6 glycosidic bonds. B. Glycogen is found in the form of granules in mammalian liver and muscle cells. Figure 2.2 is a diagram of a part of a molecule of glycogen isolated from a glycogen granule. Explain how the structure of glycogen is related to its function in cells. So, as I mentioned earlier, the function of glycogen is to act as an energy storage, and for it to act as a storage molecule, it has to be unreactive and non-soluble in water. Because you see, glucose on its own is very soluble in water, which means it can get dissolved and spread throughout the body. Not only will this disrupt the water potential of the blood and cells throughout the body, it will also be able to react with in a reaction pathways where it's not intended. So, to prevent that from happening, the body stores extra glucose, which it's not currently using, in the form of glycogen, which not only is it insoluble in water, it also doesn't react in unnecessary reaction pathways, which makes it an ideal storage molecule. C. Explain how the arrangement of cellulose molecules in plant cell walls is related to their function. The function of cellulose molecules in plant cells is to provide the cell with turgidity, or in other words, it's supposed to keep the plant turgid when the plant cell has a lot of water inside of it. Plant cell walls are supposed to prevent the cell from bursting when water content will push outward on the cell. So, you're supposed to explain how the structure of cellulose molecules inside plants allows it to resist against this pressure. In plant cell wall, the cellulose molecules lie parallel to each other, and the large number of hydroxyl groups in the polymer allow the molecules to form hydrogen bonds with water. Okay, so let me show you these hydroxyl groups. Here you have many hydroxyl groups in just one monomer, and as you can imagine, when you have multiple chains of cellulose molecules lying close to each other, all of these hydroxyl groups will allow formation of many hydrogen bonds, not just among themselves, but also with water. And on top of that, many cellulose molecules cross-link or form covalent bonds together to form microfibrils, and these microfibrils are held in fibers by hydrogen bonding. Again, hydrogen bonding will strengthen the structure of the cell wall, and this will result in a high tensile strength. This is important as, again, cell walls need to withstand the turgid pressure and prevent the plant cell from bursting. Figure 3.1 shows some fruits of the grape wine. Sucrose is transported in the phloem of the grape wine to the fruits. So phloem is one of the two transport vessels that you find in plants. Sucrose is hydrolyzed by the enzyme sucrase, which is found in cell walls. The glucose and fructose produced by the hydrolysis of sucrose passes through these membrane proteins known as hexose transporters into the cytoplasm of fruit cells. Okay, so let's imagine this is a fruit cell over here. Here in the cell wall, you will have a sucrose as well as sucrase enzyme, or let me just show the catalyzed reaction over here. The sucrase enzyme will catalyze the conversion of sucrose into glucose and fructose, and then these will pass through a transporter protein, right? That's what we have here, hexose transporters, into the cytoplasm of the fruit cell. Okay, part 1a, state why membrane proteins are required for the movement of molecules such as glucose across cell surface membranes into the cells. So let's look at the structure of cell surface membrane to understand the answer of this question. The structure is a phospholipid bilayer, so it's made up of phospholipids, right? The structure of phospholipid has a polar phosphate head pointing towards the exterior of the cell as well as towards the cytoplasm, right? These are polar heads. And then you have the hydrophobic or non-polar interior of the membrane, which will not allow polar molecules like glucose and fructose to pass through them. It will only allow small molecules like water and non-polar molecules like carbon dioxide and oxygen to pass by diffusion through them. What you'll need for polar molecules like glucose is a transport protein which will provide hydrophobic or polar pathway for them. Researchers investigated one type of hexose transporter known as VVHT1, which is found in the fruit cells of this plant. They used a mutant strain of yeast that has very few hexose transporters in its cell surface membranes to investigate the properties of this transporter. The researchers inserted molecules of VVHT1 into the cell surface membranes of the mutant strain of yeast. Equal volumes of mutant yeast cells with VVHT1 were kept in different concentrations of glucose solution. Okay, let's see if we can put this situation in front of our eyes. Let's say you've got different beakers with different glucose solution. Let's say this is 0.1 mole per dm cube, and here you've got 0.15. This could have 0.2 and then 0.25 mole, just for demonstration purposes. The rate of, okay, so other than glucose, you also have yeast cells with VVHT1 transporters. And remember, these transporter proteins will allow uptake of glucose from the solution into the cell. Okay, the rate of uptake of glucose by the yeast cells in each solution was determined. All the solutions were kept at the same temperature and pH. So these are controlled variables. The results are shown in figure 3.2. Here you can see as the concentration of glucose increases in micromole per dm cube, the rate of uptake of glucose increases up until around 150 micromole per dm cube of glucose, and after that it will start to plateau. What this is indicating is that the cells have a limited number of transporters. Okay, let's say in the 0.1 mole per dm cube solution of glucose, you have few molecules to start with that will be transported through the transporters into the cell. As you increase the concentration, you will have a greater number of glucose molecules that will be able to enter through the transporter at the same time, which increases the rate of uptake. And then you will reach a point when you continue to increase the glucose concentration, you will reach a point when all of the transporters, the VVHD1 transporters, have been occupied. At this point, no matter how much you continue to increase the glucose concentration, since all of the transporters have been occupied, the glucose molecules will enter at the same speed, which is how the rate of uptake will plateau. B1, the researchers conducted that VVHD1 is responsible for the facilitated diffusion of glucose into the cells. So facilitated diffusion is the diffusion of molecules that uses transport proteins. You're supposed to explain how the results in figure 3.2 provide evidence to support this conclusion. Well, if this were simple diffusion, then increasing the concentration would have meant the rate of uptake of glucose would have continued to increase even beyond 150 micromole per dm cube, because you would have needed no transporters. But because you need transporter for this movement of glucose into the cell, that means the maximum rate of uptake of glucose depends on the number of transporters available. Once all of the transporter proteins have been occupied, increasing the concentration of glucose will not increase the rate of uptake, and that's shown by the graph over here. The graph will plateau beyond 150 micromole per dm cube, and that's your evidence. Part 2, the researchers thought the grape vines could be modified to have more hexose transporters to increase the size and quality of grapes. So here they are suggesting that once sucrose has been converted to glucose and fructose, it needs the transporter proteins to move into the cytoplasm. If you increase the number of transporter proteins, that means, of course, a greater number of glucose and sucrose molecules can enter the cytoplasm, and it can then be tasted by the person who will ingest this grape. Explain why increasing the number of hexose transporter could be commercially important to growers of grapevines. Well, this means the fruit will turn sweeter, and this will benefit the grower as the grapes will increase their profit. Obviously, consumers will want to purchase product that seems to provide more value for their money. See, figure 3.3 is a diagram of a protein in the cell surface membrane of a macrophage from a mouse. Macrophages use these proteins in antigen presentation. Non-self antigens bind to the protein and are involved in the activation of specific T lymphocytes during the immune response. State what's meant by non-self antigen. Non-self antigens are the proteins or glycoprotein molecules usually found on the surface of any particle. In this case, this will be the foreign particle, which will stimulate an immune response. So the reason the body doesn't normally attack its own cells is because the antigens found on the cells of the body are recognized by the immune system as self antigens, whereas non-self antigens will immediately activate immune response. This is why in autoimmune diseases, the body will attack itself because it loses the ability to differentiate between self and non-self antigens. Part 2. Some pathogens enter human cells. Macrophages partially digest these pathogens and present antigens to T lymphocytes during immune response. With reference to figure 3.3, explain how T lymphocytes respond to infection by a specific type of pathogen. Okay, let me try to summarize the immune response carried by phagocytes and lymphocytes when a foreign particle enters the body. First off, the immune system has two types of cells, or two major types of cells, which are phagocytes and lymphocytes. Phagocytes are then divided into macrophages and neutrophils. You can differentiate between them by not only their function but also their appearance. For now, I'll just describe their functions. So, neutrophils will completely digest pathogens into harmless products, whereas macrophages will partially digest pathogens and display their antigens. They'll display their antigens on their own cell surface membrane to stimulate immune response from lymphocytes. Now, lymphocytes also have two categories. You'll have T lymphocytes and B lymphocytes. B lymphocytes will produce antibodies, and this is just to summarize. You have the entire process of clonal selection and then clonal expansion. Once B lymphocytes identify complementary antigens, and then you'll have T lymphocytes, which are again divided into two types. You'll have helper cells and killer cells. Killer cells will directly eliminate by attacking and destroying the cells which display foreign antigens. So these are cells like macrophages, whereas helper cells will activate they'll activate B cells to secrete antibodies. In addition to this, helper T cells will also stimulate macrophages to carry out phagocytosis faster. Now this is a summarized version, and let's see what we have to answer in this part. With reference to figure 3.3, explain how T lymphocytes respond to infection by a specific type of pathogen. Okay, so here you can see that in figure 3.3 there is an antigen binding region in the cell surface membrane of macrophages. You are supposed to relate the role of macrophages to the role of T lymphocytes. So T lymphocytes, once they are formed in the bone marrow, they will have receptors complementary to, so each T lymphocyte is specific to a particular antigen. What that means is they'll have receptors on the cell surface membranes which are complementary to specific antigens. When these antigens are displayed on phagocytes, T lymphocytes will bind to these antigens, which you can see over here, and these specific T lymphocytes will then undergo clonal expansion. So they'll increase in number by mitosis, by dividing, and then they'll differentiate into helper T cells and killer T cells, which we saw earlier, helper T cells will secrete cytokines. So as I said, this was a summarized version. And there are more details that go into the entire immune response. So helper T cells will secrete cytokines, which will be the cause of stimulating B lymphocytes and killer T lymphocytes to bind to infected cells and destroy them. Here you are only supposed to explain the role of T lymphocytes, starting from starting right after they formed in the bone marrow. Question 4. The gene for enzyme catalase is on chromosome 11 in humans. A. Explain the meaning of the term gene. This is a sequence of nucleotides or bases that is a part of DNA which codes for a polypeptide. B. Two enzymes, DNA polymerase and DNA ligase, are involved in the replication of DNA. Figure 4.1 shows the replication of a human chromosome 11 by DNA polymerase. The arrow shows the direction of synthesis of the new polynucleotides by DNA polymerase. In conservative DNA replication, you have multiple steps. First, the DNA molecule, which is a helix, this will unwind into a straight molecule and then it will unzip. What that means is the hydrogen bonds between the bases will break down, so you will have two separate strands. Now the entire DNA molecule does not undergo this process at the same time. A part of the DNA would still be in the double helix form, and that's what's shown over here in figure 4.1. You can see the DNA polymerase enzyme builds the new strand from 5' to 3' direction. So it will add new bases or new nucleotides to the 3' end, and this will, as you can see, this is moving towards the part where the DNA is where you still have hydrogen bonds between the bases. This is the replication fork. So the DNA polymerase will build the leading strand, which means this strand over here, towards the replication fork, whereas the lagging strand, which you can see over here, this one is built away from the replication fork. You can see that the DNA polymerase enzyme is moving away from the replication fork. So once again, the leading strand is built towards the replication fork, and the lagging strand is built away from the replication fork by DNA polymerase. Part 1. Describe roles of DNA polymerase and DNA ligase in the replication of DNA. DNA polymerase can only build new strand in the 5' to 3' direction. Now, there is a difference between the construction of leading and lagging strand. DNA polymerase is able to continuously build up the leading strand by adding new nucleotides to the 3' end over here. However, for the lagging strand, that is not possible. On the lagging strand, DNA polymerase builds the strand in short segments known as Okazaki fragments. These fragments consist of a few nucleotides, and then DNA ligase is responsible for joining these Okazaki fragments on the lagging strand by catalyzing the formation of phosphodiester bonds to create the sugar phosphate backbone, whereas on the leading strand, DNA polymerase can build the new strand by directly forming phosphodiester bonds between adjacent nucleotides. Okay, let's say this is the leading strand. This is built completely by DNA polymerase as it continues to add nucleotides to the 3' direction. And here you can see how the lagging strand is built. DNA polymerase will build these Okazaki fragments that have a few nucleotides joined together, and then they will be connected by DNA ligase enzyme to form the entire lagging strand. Part 2 state the name of the stage of interphase in the cell cycle when DNA replication occurs. This happens during the S phase, which lies between G1 and G2 phases of the interphase. C. Figure 4.2 is a diagram of chromosome 11 at metaphase of mitosis. So this is the second stage of mitosis, as you have prophase, metaphase, anaphase, and telophase. Part 1, state the names and functions of structures A and B. Structure A here is the centromere, which is responsible for connecting together the sister chromatids in a chromosome, and structure B is these are the spindle fibers, which are responsible for aligning not only aligning the chromosomes along the equator of the spindle during metaphase, but also pulling apart the sister chromatids during anaphase. Part 2, complete figure 4.3 to show what happens to chromosome 11 in anaphase so that the daughter nuclei are genetically identical. So here you can see these centrioles, which are connected to the centromere of chromosomes by spindle fibers, and these will pull the sister chromatids apart from each other during anaphase, as the spindle fiber will contract in length, and this is how you will need to point the sister chromatids with the spindle fibers connecting the centromere to the centrioles. Figure 5.1 is a longitudinal section of a capillary in muscle tissue as reviewed with a transmission electron microscope. A. State the evidence visible in figure 5.1 that indicates the cells inside the capillary as red blood cells. So red blood cells, as you can see in this transmission electron micrograph, first of all, that you have different cells visible here. This one, and here you can see the organelles inside of the cell, whereas in these cells you don't see any of that. That's one indication. The other one is that these are biconcave in shape. Both of these are features of red blood cells, that they can bend into biconcave disc shape to be able to flow through capillaries, and they do not have large organelles like nucleus and mitochondria. B. Explain how the structure of capillary wall is related to its function. So the capillary is only one cell thick. It's made up of endothelial cells which have gaps between them. These are called endothelial pores. So the one cell thick wall of capillary provides a short distance for diffusion, allowing rapid gas exchange. This is useful especially in capillaries that, for example, are lying next to alveoli, which will allow diffusion of carbon dioxide and oxygen into and out of capillaries, between capillaries and alveoli. Also, the endothelial pores, or the gaps between endothelial cells in the capillaries, will allow tissue fluid to form, as they'll allow the contents of blood to leak out of the capillaries into the surrounding tissue. Figure 5.2 is a diagram showing some of the events that occur as blood flows through a capillary in a respiring tissue. Part 1, an increase in respiration results in an increase in carbon dioxide concentration in the blood and the release of more oxygen from red blood cells to tissues. Explain how an increase in carbon dioxide in the blood leads to the release of more oxygen from red blood cells. Okay, let's start by looking at this diagram over here. You have cell of respiring tissues where you will have carbon dioxide being produced from respiration. Okay, so carbon dioxide will move into the blood and oxygen will move from red blood cells back towards the respiring tissue. Now, the carbon dioxide which diffuses from respiring tissue will move into the red blood cell. It will combine with water and it will get converted to carbonic acid by the catalytic action of carbonic anhydrase enzyme. The carbonic acid will then dissociate into protons and bicarbonate ion. Bicarbonate ions will then diffuse out of red blood cells, whereas protons will remain inside of red blood cells, and this will be the cause of displacing oxygen from oxyhemoglobin so that it can diffuse out of red blood cells and towards respiring tissues. Okay, so to answer this question, why an increase in carbon dioxide in blood leads to release of more oxygen from red blood cells. So as carbon dioxide diffuses from respiring tissues into the red blood cells, it will combine with water to form carbonic acid, and increased carbon dioxide will result in greater production of carbonic acid, and of course this will lead to a greater production of hydrogen ions as a result, which we saw earlier. Initially, hemoglobin in red blood cells is bound to oxygen to form oxyhemoglobin, but hemoglobin has a high affinity for protons, which result from excess carbon dioxide. As carbon dioxide increases, protons increase, and due to the high affinity between hemoglobin and protons, the protons will bind to hemoglobin and they will displace the oxygen from oxyhemoglobin, which will make more oxygen available to the respiring tissues, as it will diffuse out of red blood cells towards the tissue. Now in this question, it's important that you state that due to more carbon dioxide, there will be more carbonic acid and more protons as a result, which will lead to release of more oxygen from the results, as that's what they're asking in the question. Part 2, chloride ions are constituent of blood plasma. The concentration of chloride ions in plasma of deoxygenated blood is between 2 to 4 millimole per dm cube lower than in the plasma of oxygenated blood. Explain why the concentration of chloride ions in the blood plasma of deoxygenated blood is lower than in the plasma of oxygenated blood. Here you need to understand the chloride shift. This occurs when bicarbonate ions, which you can see over here, move out of red blood cells. This entire process, their diffusion out of red blood cells, and as a result of this, chloride ions will move into the red blood cells to prevent electrical imbalance. Now, deoxygenated blood has a higher concentration of carbon dioxide, which eventually leads to a higher production of bicarbonate ions in red blood cells, as they diffuse out of the cells into the plasma, more chloride ions will diffuse from the plasma into the red blood cells, again to prevent electrical imbalance. And this is why you will have a lower concentration of chloride ions in the plasma of deoxygenated blood. Question 6. Figure 6.1 is a diagram showing the passage of water through the tissues of a flowering plant from the soil to the atmosphere. The arrows show the direction of water movement. A. The structure labeled X is part of the simplest pathway. State the name of structure X. So structure X over here is the plasmodesmata, which is the gap between cell walls, which allows movement of substance between two plant cells without them having to pass through the cell walls and cell surface membrane. This allows for a faster pathway known as simplest pathway, as opposed to the apoplast pathway, which is movement of substances through the cell walls. B. The structure labeled Y in the cell wall is a barrier to the apoplast pathway. State the name of structure Y. This is the Casparian strip, which blocks the apoplast pathway in the endodermis layer of roots. So at this point, all of water movement will be through the cell walls and cell membranes. C. With reference to figure 6.1, complete the statements about the movement of water in the flowering plant. Okay, let me try to summarize this movement by transpiration. The movement occurs when water vapor over here. You will have water vapor in the air spaces of the leaf when this diffuses out of the stomata, and this will leave the air spaces inside the leaf empty. Then what happens is water vapor on the cell walls of mesophyll cells will evaporate into the airspace, and hydrogen bonding between water molecules. So here you have water molecule in the mesophyll cell walls, and then you will have water molecules in the xylem vessel as well. So hydrogen bonding will draw water out of xylem into the cell walls after water has been evaporated from cell walls of mesophyll cells. This will pull up more water in the xylem vessel by transpiration pull, and this has resulted from hydrogen bonding. So let's see if we can now complete this part. Water moves from the soil solution into the cytoplasm of root hair cells by osmosis, because this is movement of water down the water potential gradient. The amount of water in the soil particles is higher than that in the root hair cells, and then water moves from the xylem into the root, from xylem in the root to the leaf by transpiration pull, as I mentioned. This results from hydrogen bonding between water molecules in the cell walls of mesophyll cells and water molecules in the xylem vessel. Okay, and then water moves from mesophyll cell walls into the air spaces by evaporation. So this is evaporation of water from cell walls into the air spaces of the leaf, from where the water vapor will move from the space to atmosphere outside the leaf by diffusion. And this is it. We are done with this paper. Thank you for watching.